Well, how are you? All right. Welcome. So, did you want to give an update or discuss some things going on with you, or?
Yeah. Last week I did some analysis on the lineage information which I have received from that thing, and me to work on certain things like like the temporal, like the time where the synthesis has actually evolved. So I've looked into some of the analysis, and I just did some, the basic analysis, seeing like where at what time point synthesis have been formed, and how many of them have been formed, and what is the frequency, where it actually lasted. So I was able to look into some of the details as that. So that's kind of it from my side.
Okay. Hi.
Hello.
Yeah. Well, that's good. Thank you. All right. So, does anyone else have anything? I don't know if you've been working on the syncytial stuff. Did you want to talk a little bit more about that?
Well, I came across a weird paper.
Okay.
As an animal grows, its muscles, multinuclear muscle cells, get bigger, and then they divide in two.
Okay.
Okay. So it's interesting. They did not look at the nuclei, unfortunately. So we don't know if there are more nuclei or if the nuclei divided in two.
Yeah.
If they doubled.
Right.
Okay.
Yeah.
So it's a curious fact. There's something about they were looking at muscle cells in mammals, I think, and it's a curious thing that a syncytial cell might behave in terms of cell division much like a normal cell.
Yeah.
Okay, but until somebody does the work, we won't know.
Right.
Lalith, I think, has all the information. Bradley, what we're trying to do that's different from what you do is put all of the formation and successive steps in syncytia on the same time scale as the previous lineage tree.
Right.
Okay, you have the data there, but they're organized horizontally rather than vertically.
Yeah.
Okay, so that's what we're looking at, and he's got all the data and corrections from the guy who did the interactive lineage tree.
Yeah.
So I think we can go ahead. We can go ahead now and make a definitive, and how should I say? It should replace this other fellow's tree. The only question remaining is whether or not he wants to be a co-author on the paper. So, it's basically a correction and addition to his interactive lineage tree. Well, Layla thinks it's best to start from scratch rather than get his code.
Yeah.
I don't understand why. Layla, can you explain that?
So probably what he actually feed it into the website or the backend. So the databases that he used in the previously, he did it manually from scratch. When he uploaded into the system, so he collected all this data and he has given to us. I think when we have all the data, I would prefer the platform which I use currently to extract the insights from it, rather than taking his the older versions of the technologies, and so it's more like quite old. So it's like more like HTML, and backend they used. I don't really know details now. We use, and the visualizations and the kind of the user interfaces have been improved a lot in time, and they're evolving rapidly. So I think the latest could be good and cool.
I got you. Okay. Okay, so it's better to start over.
Yeah.
Okay, so that's where we're at. The Syncytia are everywhere. The paper includes a large list of reports of Syncytia.
Yeah.
So, and this curious paper. Okay, I'll look up this paper now. I'll shut up and look it up.
So Susan, are you? What's going on with you?
Sorry, I turned the mic off because
Oh, there you are. You're in.
barking, and there's nothing wrong with my microphone.
Okay. Okay. we can hear you.
I'm just busy trying to get you
This paper attributes the muscle growth to mechanical tensions. Okay.
Oh, okay, that sounds good.
Now, of course, this is post-embryonic.
It's
Right.
what?
Post-adult stage or childhood stage. I'm not sure. Anyway, it was a surprising result. I don't know. Do nematodes do this?
No, I don't. In fact, yeah, the body wall muscles aren't Syncytia. Yeah, they're mononucleate cells.
Okay. Nobody's tried to stretch a nematode.
No.
You know how to do it.
Yeah.
You can put it in a capillary tube and then use water flow to stretch it in two directions in a T-tube.
Yeah.
You put the water down, and if you don't flush the nematode out, you're stretched. So it would be possible to apply forces to a nematode and see how it responds.
Yeah.
Okay. There some of these mechanical probes, they're light probes that exert forces. So it might not be out of the question to stretch a nematode.
Anyways, I'm just randomly looking up things so that I can write a paper, and I've done the summary of my thesis, but he wants me to do a literature review.
Oh, okay. Yeah.
So this fits in with it. Like, why am I looking at this, and why is it important?
Yeah. Well, we can take a look at these three. I think it's three, right? Or it's four.
Yeah.
Oh, we'll go over those. Yeah. Okay. So yeah, let's look at those papers. So the first one is the one Dick put in the chat. This is muscle growth by sarcomere divisions. So this is from this year. So that's good.
Oh yeah, that's why I can't follow it up and find out.
Yeah.
Nobody's done any work, I presume, at this point.
Yeah. So this is on muscle growth in, I think, humans, or no, Drosophila. This is so this wouldn't necessarily be relevant to C. elegans, but let's see what they have to say.
You never know.
Well, let's see. The sarcomere is the elementary contractile unit of muscles. Adult muscle cells are large and chain thousands of sarcomeres into long periodic myofibrils that attach to the skeleton. During development, muscle cells must increase in length to maintain the mechanical connection to the growing skeleton. So they have to stretch out as the skeleton grows and everything else. And they have to kind of maintain that sort of mechanical connection, but also the same tension that they maintain, as they grow, as the entire body grows. How muscles add new sarcomeres to facilitate muscle growth is unknown. So they need to add in sarcomeres. Using live imaging and high throughput image analysis, we have now tracked the sarcomere components during the developmental growth of Drosophila muscle and found that individual sarcomeres divide along the myofibril tension axis into daughter sarcomeres. So this is where you have this tension axis, and this is where you're getting this division of the sarcomeres into these, basically they're replicating. This way, new sarcomeres can be inserted into the contractile, mechanically intact myofibrils. There's remodeling going on, but it has to be incorporated into the existing muscle. kind of poses an interesting problem, which is the organism is constantly using its muscle to move, but it's also needing to remodel it continuously. I mean, muscle tends to remodel continuously because it gets damaged as it's used, and so there's that aspect, but also there's this other aspect of developmental growth. We propose that sarcomere division is triggered by tension and local sarcomere damage. This is what I was referring to, originating from skeletal growth and muscle contractions. Sarcomere divisions repair damaged sarcomeres, ensure their mechanical integrity, and synchronize sarcomere addition with skeletal growth during animal development. So it looks like this is figure 1 is an image of kind of what they're dealing with here. This is the Drosophila sarcomere scheme. So this is similar to the sort of mammalian sarcomere scheme. You don't, you have the I bands here. You have the Z discs over here at the ends, and you have the A band and the M band. So you have this sort of structure where it's being stretched and pushed together at the ends, and then you have this middle part that does the stretching. So you have actin filaments in here, you have myosin in here, and you have obscurin in the middle. So that's the way that our muscles are structured as humans, and as you see the same structure in Drosophila sarcomeres. Now these have to be replicated in development. That's what they're referring to. So you have to have this division, and you have to replicate the structure within the cell. So this is then flight muscle length growth. So this is where you get this the muscle cells, and they look like they grow outward. You have these tendons that are connected, and so these are the flight muscles and how they're expanding outward like this. And so this is some staining here showing this flight muscle growth. So you go from 32 hours where you have sort of this, and you see this down here in the stain. You get 36 hours, it gets stretched out. 40 hours, it gets stretched out more, and then 48 hours. This is the 40-hour example, and then this is 48 hours. So you can see that it stretches out quite a bit, and it's larger, more robust. And then D is the stable connection of the flight muscles to the tendon cell extensions. Green lines during the growth phase from 32 to 40 hours after puparium formation, so post-puparium. So this is again where you get these banding here, and you can see the variation. And then E is the sarcomere addition rate. So how do we keep adding sarcomeres to the structure? This is the muscle fiber length on the y-axis, and you have time on the x-axis. And then sarcomere length is function of muscle fiber length. And then the sarcomere addition rate is a function of that as well. So you see that you have the sarcomere length function, which is that it's sort of stable across time, but you get the sarcomere addition rate here, and then you get this muscle fiber length growth here. So this is kind of showing this.
Is it clear if the new sarcomeres are end to end or parallel?
I'm not sure.
Okay, that's one thing to try to eke out of this.
Yeah.
You see, I found nothing about nuclei in here.
Yeah, they don't really talk about that. Yeah. So I guess this is flight muscle end growth, and this is live imaging of this sort of the ends here. So you can see up here at the top, you have these ends of the flight muscle connected by the tendons. So you have the tendons connecting into the muscle, and they just image the end, and they want to see what happens during growth. So you see that there's this, they're using this GFP reporter, and it stretches out over 180 minutes, 23.2 microns, And you can see this here zoomed in where you have the Z bands that are identified here, and they get stretched out like that. Yeah. And then in figure 3, they just talk about their automated detection tool. Figure 4 is sarcomere detection reveals sarcomere division by myosin segregation. So again, you're getting these sarcomere divisions by the segregation of myosin. You can see in here we have these different molecules that are stretching out as this process happens. Sarcomere division into 2 daughter cells. So I'm trying to figure out what's going on in this figure. You have actin, thalamus, obscurin, myosin, and let's see if I can zoom in a little bit more. And myosin. So those are the 4. And then each of these graphs kind of show their difference between Z1, M, and Z2. So the Z band 1 and Z band 2, and then M in the middle. And so you can see that the obscurin increases in the middle. Actin is maximized at Z1 and Z2, thalamus as well. And then myosin is also in the middle with obscurin, but there's a little bit different profile to it. And you can see as you move across the band, you see that there's this difference in these molecules. So that suggests that there's this sort of segregation of the sarcomere into 2 sort of cells, that these molecules are moving towards the ends. You can see by the time you get to this part of the band, you can see it. This is weird. This part of the band, that the profile is quite different than at the beginning. So you see this in the stain here, where you have this difference in how the molecules are distributed and so forth. So that's what they're showing there. And then this one here is live imaging of sarcomere division. So this just shows over 14 minutes kind of what we have here. So we have these images where things get farther apart. Yeah. And then this is the larval sarcomere Z discs in 3D. So they're doing some 3D modeling of this microscopy image. So they have the Z1, the Z2, and the Z3. And they're just showing in this sarcomere Z discs in situ. Right. So it gets interesting. They have a discussion here. They talk a little bit about sort of how, so they identify a sarcomere division mechanism that allows the addition of new sarcomeres into existing myofibrils anywhere along the growing muscle fiber. This is fundamentally different than their end hypothesis, which they started with, which proposed that new sarcomeres are exclusively inserted at the terminal Z discs of muscle fiber ends. So instead of being inserted at the ends and then pushing everything over, they kind of insert wherever they can into the scheme. And so the latter may still apply under certain circumstances, which by the latter they mean the end hypothesis, but would require the transport of all newly synthesized sarcomere components to the muscle fiber ends, and the damage to be limited to the ends. Okay, and then they talk about sort of how these additions are regulated. Every sarcomere division requires the controlled breakage of protein-protein interaction bonds without an uncontrolled break of the entire myofiber. We propose that controlled damage is triggered by the high mechanical tension, which is built up by skeletal growth that stretches the attached muscles while they keep contracting. So there's this tensile force that's managing this growth process. So as the organism uses its muscles and as the organism is growing, there's this tensile force that sort of triggers this process. So, and then when growth stops, there's still damage to the muscles that's being used, but it's less, it doesn't trigger this process. As a consequence, tensile forces in the sarcomere increase homogeneously along the myofibrils until a threshold is passed and a sarcomere division is triggered, where the zipper progresses forward. So that's kind of what their hypothesis is here. And so this division initiation creates free binding sites that allow the recruitment of new sarcomere proteins, making the division process irreversible. During the entire process, myosin can still interact with actin filaments, which prevents the uncontrolled rupture of the entire microfiber. Last, the centrally located thalamus, actin, and Z-disc components establish a new Z-disc between the segregated myosin stacks, and then establishment of a new Z-disc. So they show the sarcomere division model in figure 7, and they show that there's this structure where you have what we saw in the first figure. We have the ends and the central part. So you have the I bands and the A band in the middle, and you have this high tension outward. So the tension is coming from the organism growing, and it's stretching the sarcomere out. This tension builds up. You get this division initiation, and that releases the tension because there's something new being formed in this space. So it expands the space and it lessens the tension. So you see that this is kind of this I band and the A bands are here, and they get stretched. And then eventually you get a new I band, A band, I band combination. And then that releases the tension because there's more there in the middle. And so that's what we have here. But to do this, to have this division, you need novel protein recruitment, and then you need to actually establish a new Z-disc altogether. So this is where you get this. Now you have two of these I band, A band, I band structures with a Z-disc separating them, and now the tension is lower. And then that process will repeat as the organism grows further, and it stretches on it some more. So that's that paper. That's an interesting paper kind of talking about how these structures get formed. Now, in C. elegans, we don't have this kind of structure for muscle. They're not syncytium. They're body wall muscles, so they're independent cells. They divide and then form this body wall. And there are a lot of syncytium around it, but they're basically, I think the cells just grow in place and then start to exhibit twitching, and then they start to work together later. But this is in Drosophila. In Drosophila, you get a model that's similar to what you see, say, in mammals. So it's interesting. Yeah, they don't talk about the, they talk about the part about the sarcomere divisions, and it's just basically guided by this tension that's happening as a consequence of growth. So, yeah, and I don't know, like, it's interesting, some of the syncytium you see in C. elegans are those driven by similar forces. So in other words, we saw last week where you have this process of these developmental cells forming precursor cells that fuse, and then you have rounds of nuclear division, and you have this syncytial structure that's growing with the organism. And it's growing kind of in place along. The one that we saw last week was assembled, kind of in the larval development. So the worm is out of the egg. It's moving around. It's experiencing tension. And so, what is the role of tension in that building that structure? I'm not sure. It's a deterministic structure because you see it in the lineage tree as a consequence of the lineage tree. And it's basically the same in every C. elegans. But, it's got to kind of grow with the organism and be built as the larval organism is behaving and growing in size. Okay. Do we have any questions on that? So let's get to the paper Susan brought up. So the first one here is this cytoskeletal prestress. So this is in Cytoskeleton Journal in Hoboken, and this is from 2021. So this is um, something they call cytoskeletal prestress, the cellular hallmark in mechanobiology and mechanomedicine. So this is a review paper. So basically, their hypothesis is that increasing evidence demonstrates that mechanical forces, in addition to soluble molecules, impact cell and tissue functions and physiology and diseases. How living cells integrate mechanical signals to perform appropriate biological functions is an area of intense investigation. Here we review the evidence of the central role of cytoskeletal prestress in mechanotransduction and mechanobiology. Elevating cytoskeletal prestresses increases cell stiffness and reinforces cell stiffening, facilitates long-range cytoplasmic mechanotransduction via integrins, enables direct chromatin stretching and rapid gene expression, spurs embryonic development and stem cell differentiation, and boosts immune cell activation and killing tumor cells, whereas lowering cytoskeletal prestress maintains embryonic stem cell pluripotency, promotes tumorigenesis and metastasis of stem cell-like malignant tumor repopulating cells, and elevates drug delivery efficiency of soft tumor cell-derived microparticles. So basically, the way they envision it is that it controls a lot of different things in terms of growth, in terms of stresses on the organism as it's growing, and then some things having to do with cell differentiation, whether that be stem cells or cancer cells. And, there's a lot of mechanical stuff going on there that sort of informs, especially in mammals, what the cell might become, or, whether, say, cells adhere to a surface or what have you. So it's really interesting. I mean, they're kind of saying that it controls a lot, that this phenomena is the root of a lot of things in cell biology and in development. The overwhelming evidence suggests that the cytoskeletal prestress is the governing principle and the cellular hallmark in mechanobiology. The application of mechanobiology to medicine or mechanomedicine is rapidly emerging and may help advance human health and improve diagnostics, treatment, and therapeutics of diseases. So this again is a review article. They kind of give some figures here. They just show one here of the different cytoskeletal prestresses, and this is just kind of a summary of what they're talking about. So this is where they talk about stem cells, malignant tumor cells, mechanotransduction, and immune cells and microparticles. And so in this stem cells example, you have prestress-mediated fate determination. So we have, of course, this phenomenon called pluripotency or multipotency, whereby a stem cell or a precursor cell has a number of fates that can go down. Like you have muscle precursors, you have neural precursors, and you have stem cells that are sort of, muscle stem cells or neural stem cells. And so at every step, they have this set of fates that they can achieve. And in, say, like in regulative development, which we don't talk about much with C. elegans, there's some of that. But in those cases, you have this sort of these cell fates that are controlled by chemical cues. And what they're arguing is that mechanical cues also play a very important role in achieving these fates. So they have prestress here, which limits or determines the fate, or at least partially. And so that's the case for stem cells. For malignant tumor cells, you have this malignant cell softness, where you have tumor cell self-renewal, and you have these prestresses which enable this process or retard this process. Then we have mechanotransduction, So we have this prestress, and again with mechanotransduction, prestress actually increases the range of things going on. So you have anisotropic gene activation, chromatin stretching and gene upregulation,
Another pair.
long-range stress propagation and enzyme activation, substrate stiffness sensing, and cell stiffening. So as you get this pre-stress mechanotransduction increases. And then with immune cells and microparticles, you get this pre-stress mediated immune cell activation and killing of tumor cells. You have a microparticle uptake function, and you have a microparticle release function. So as you can imagine, if the uptake is where you increase the number of microparticles, the release is where you get rid of them, and then that affects the pre-stress affects these. So as pre-stress increases, uptake decreases. As pre-stress increases, release increases. That sort of thing. And then this is their sort of diagram of how this works. So you get these external stresses, and this is the membrane here, plasma membrane. You have these different things happening at the membrane. You have growth factors, signaling cascades being induced. You get these ion channel mediated things with piezo1, which is a ion channel that explicitly involves forces and sensing a mechanical phenomena. You have cadherins and other things at the membrane, and then those signals get propagated into the nucleus. Actomyosin pre-stress comes from these things that get propagated from the membrane down to the nucleus. Then the nucleus, of course, is affected by chromatin being stretched and then genes being activated. So you get these forces that are communicated into the cell through the membrane. They trigger processes in the nucleus that allow for gene activation and activating muscle stress-dependent cell or genes and other types of processes. So that's the hypothesis they have there. So this is a nice review paper. And then we have this other paper that Susan posted from Biophysical Journal. This is mechanochemical signaling, direct cell shape change. So this is very similar in terms of the model, that mechanochemical signaling is doing this work to affect the cell, its its phenotype, and in this case, cell shape change. So they're attributing this mechanochemical signaling to they're attributing at least part of that effect to mechanochemical signaling. So this is the abstract for specialized cell function, as well as active cell behaviors such as division, migration, and tissue development. Cells must undergo dynamic changes in shape. To complete these processes, cells integrate chemical and mechanical signals to direct force production. This mechanochemical integration allows for the rapid production and adaptation of leading-edge machinery in migrating cells, the invasion of one cell into another during cell-cell fusion, and the force feedback loops that ensure robust cytokinesis. Quantitative understanding of cell mechanics, coupled with the protein dynamics that exist, has allowed us to account for furrow-ingression during cytokinesis, a model cell shape change process. At the core of cell shape changes is the ability of the cell's machinery to sense mechanical forces and tune the force-generating machinery as needed. Force-sensitive cytoskeletal proteins, including myosin II motors and actin crosslinkers, accumulate in response to internally generated and externally imposed mechanical stress. So this is, where you're getting shape changes and things like that, where you're getting activity within, say, the cell that's affecting shape, but also you're getting these external stresses and external forces, and those are kind of contributing to this. The physical theory behind how these proteins display mechanosensitive accumulation has allowed us to predict paralog-specific behaviors of different cross-linking proteins and identify a zone of optimal actin binding affinity that allows for mechanical stress-induced protein accumulation. So they're interested in paralogs, which are these types of genes that have a specific evolutionary relationship, and they're hypothesizing that this is something that you see widely across species. These molecular mechanisms, coupled with the mechanical feedback systems that exist in cells, ensure robust shape changes. But if they go awry, they are poised to promote disease states such as cancer cells, metastasis, and loss of
What?
tissue integrity.
Oh, what do you mean? A set of papers. I don't know exactly.
That's the cell space splitter. They just described it.
Can you put that reference in the
Yeah,
chat,
it should
please?
be the second one that Susan posted.
The second.
Yeah, well, let's see. Up here, that's maybe not. There's a lot of stuff in the chat all of a sudden. This is, let's see, mechanochemical signaling direct cell shape change. So it's this one here by Schiffhauer and Robinson, 2017.
Okay.
What species?
I don't know if there's a specific species. I think this is just kind of a general model that they're describing. So this is the sort of their first figure. This is where they talk about the structure of cortex, determining the shape. And so this is, I think, generally across species. Yeah, they don't talk about a specific. Well, they talk about things like dictyostelium, but that's like a general kind of like these are. And then they talk about mammalian cell types. So I think it's a general kind of approach to this.
First they tell you you're wrong, then they tell you it isn't important, then they tell you we always knew it.
Yeah.
Schiffhauer and Robinson?
Yeah, that's what this is.
Okay.
But yeah, I mean, it's yeah, I think it's just kind of a general kind of theory in animal cells, of course.
I'm off for coffee.
Yeah.
Delightful. I'm glad to have heard that. That was
Yeah.
fortuitous.
Yeah. So it looks like we have a lot of stuff in the chat here that I didn't see. So let's go over that. Yeah. Well, this is Hildebrand, Holton, Subramani, etc. This is from 2017. I guess this is Susan as well. The E-cadherin and modal 2 complex organizes actin filaments required for the epithelial hexagonal packing and blastocyst attaching. And again, it's probably a more general paper on development. So that's again this very specific model of actin filaments in cells. Then there's this paper here, stem cell mechanoadaptation 2, microtubule stabilization and substrate compliance effects on cytoskeletal modeling. This one here is, I guess it's a 2, so there's probably a mechanoadaptation 1. Okay, but that's okay, so that's that paper. Then there's negative tension and control stability and structure of intermediate filament networks. So this is about negative tension again. And then this is shearing cells. This is a paper from Dick and Natalie and colleagues.
Yeah, this is how to stretch a nematode.
Okay. Sheared drops in pennate diatoms. So this is in diatoms where they're doing sheared droplets. What is that? What were the lessons from that paper?
I heard a talk on, actually it was on paint drops that are put into a flow, which is coming up in the water, and then it stretches in one direction and compresses in the other. So you can actually change the shape of a drop that's suspended at the point in the middle. Okay, so it's a very gentle stretch and compression, making the thing elongated. And I made that an analogy with diatoms that are in flowing streams.
Okay.
Okay.
Yeah.
So it's just an analogy at this point, but the flow pattern would allow one to try to stretch a nematode. You could put a nematode in there and apply gentle stretching forces to it and see what happens.
Right.
Because it's already elongated, so it should orient nicely in the flow.
Right. Yeah.
Okay. And I put another one in here. I think it's further down. This is where we compressed axolotl embryos. And we got a weird effect. If you compress them, I think, for 6 hours, you put between two glass plates, they end up arched backwards. In other words, I'm not sure how to illustrate. This is the head, and this is the tail, and the whole embryo is shaped. It's concave.
Right.
Okay, so it was an interesting observation. We didn't do anything more than publish this abstract. I put the whole abstract in there.
Right. Yeah. The whole, the text of it. Yeah.
Yeah, the whole text of the abstract.
All right. Yeah.
It was a curious result. I don't think anyone's repeated it, but it's called kyphosis. I think babies are occasionally born with this defect, very rarely, but it's a major defect of an embryo,
Right.
simply caused by compressing the embryo for a few hours.
Yeah. Yeah, that's like the paper we saw for the inducing of syncytium in C. elegans, where you have you compress the embryo and you can get syncytium.
It's also fits in with what I saw with my axolotls that I crossed, two morphos, and they produced little hatchlings that were bent 90 degrees. The tail was bent 90 degrees from the body. And they had a very small and
which direction?
Blastocyst.
Was it bent left, right, or up, down?
Left, right.
Oh, that's peculiar. Okay.
Yeah, well, they were weird. They didn't survive. The only one that really survived was the foreshortened one.
Okay.
They named it Squishy.
Squishy.
He named it Squishy. This is Elizabeth.
Yeah, I think all of these papers are tending towards solving a problem which is outstanding in embryology, and that is, how is the shape of the embryo formed?
Yes. And I'm pretty sure axolotl, it depends on the blastocyst.
Oh yeah, it's probably a cumulative effect of all the stages, but we have theories of differentiation, but we don't really have proper theories of morphogenesis. Okay, and I suspect one could get at this by measuring the forces and the stretch and the how should I say, the strain, stress and strains in embryos. The strain is easy to measure because it's just the distance between two cells changing over time. Stress is hard to measure because you got to get something in there and measure the force. Right.
Yeah, or you have to know the elasticity of.
You do all sorts of, do a proper mechanical analysis. But strain is easy to measure.
Yes, it's just how much it stretches.
Yeah.
Yeah.
Yeah. So if we could ever get a strain map over an axolotl embryo during development, might be able to make some headway with this.
Yeah.
Now you'd have to use the atomic force microscopy.
No, you just have to be able to identify 2 cells anywhere in the embryo.
Oh, you just want to do the strain. Oh, yeah. Yeah.
Strain. Yeah, you just have to identify, so you can follow 2 cells, and then you can make a strain map. If bigger, you can do a strain map over the whole embryo.
Okay. Yeah.
Okay, so it's approachable but difficult.
Yes. Unfortunately, the little microscopes I've got in the ball microscope are not decent resolution.
Oh, you can't track cells with them.
Well, you can, but they end up being blurred, and like there's no depth of field, and they're like 3 megapixels maybe. Like I'd like them to be a 14 megapixel camera.
Oh, okay.
Yeah.
You have to get that new telescope, which does how big an image? Many gigabytes.
Yeah. Yes. I need something like that, and it's and nobody's thinking small. Oh, we have to look at the universe.
Yeah, well, it'll only pick up 1 billion galaxies at once.
Yeah. I want a few axolotl cells.
Yep.
Oh, well.
Yeah,
Anyway,
one thing we used out of desperation was to do pseudo color. Okay. You take your grayscale image, turn it into a pseudo color image, and then look for color patches moving. Another thing is to use embryos that are lightly pigmented so you can follow pigment patches.
yeah. I'll just have to persuade Cytoskeletal to sell me some of their interesting breeds.
Oh, okay.
Yeah, they have one that's sort of a brown mottled
Oh, really?
mottled one, and I wondered what its eggs would look like.
Yeah. Yeah, we used white, I think.
Yeah, white leucistic. They call it leucistic.
Yeah, they have like
white, white skin, black eyes.
Yeah, I think that's
right. You can see the cells better with those. And that's what I've got, actually. I've got a pair of those right now.
Oh, good.
Yeah. So, but that only gives me 1 batch of eggs. So I hope they survive this summer. But anyway, I'm still trying to approach Cytoskeletal, as in Saskatchewan axolotl, only they shortened everything. Cytoskeletal. Anyway.
Anyway, I think, as I said, I think all these papers are tending towards solving that problem. It's how does an embryo pre-stress? How does the stress affect morphogenesis and differentiation? These are all unsolved problems right now.
Yeah. Anyways, I'm researching this for my modeling paper, and I'm hopefully going to get written this summer. Anyway, yeah. So.
Yeah. That's great. Thank you. Yeah, thanks for the papers. So that, and I'm glad that they all had a theme. All right. Well, I think that's it for today. Thanks for attending.
let's see.
I should try to come up north and visit you, Dick.
Oh yeah, we're far north. Okay. Yeah. Oh, here, let me throw one more paper in.
Okay.
You may have already seen this. I'm not sure. Where is it here?
Oh yeah, we talked about this, I think, in a meeting.
Oh, yeah. Okay.
Yeah. It's a good paper. We were talking about it.
Still waiting for Mike Levin's reaction, but
Oh, that's supposed to be like a response to Mike Levin's work, or
oh yeah, exactly. I've been trying to get him to write this paper for decades.
oh, okay. Thanks. Thanks for
Okay.
attending. All right. Bye.
Okay, thanks. Bye.
Okay, I wanted to mention a few other things for today's meeting. The first is that on Friday, we got an update on our Google Summer of Code projects. And Jayadratha, who is working on DevoGraph, and he's working on these temporal graphs, he's been exploring Connectome data. And for C. elegans, you have a lot of nice open source Connectome data that people collected. Different types of data sets, as we'll discuss, that characterize different aspects of the C. elegans connectome. So they could be the chemical connections of synapses, the electrical connections of gap junctions, the male connectome, the hermaphrodite connectome, the extrasynaptic connectome, and we'll talk about those in a minute. But this is a resource from openworm.org. It's called the Connectome Toolbox. So this is hosted by OpenWorm and was built by, I think it was Poré Gleason's group at UCL, University College of London. And so this overview kind of collects all of the open data sets that exist and puts them together in one place, and then shows kind of the relationship between these data sets. So let's go down here. So this shows kind of what you can do with these data. As you may know, gap junction connectomes of C. elegans look slightly different than the connectomes that involve synapses or these chemical connectomes. You have extrasynaptic connectomes like neuropeptidergic connectomes, and those are going to look different still. So you can represent connectomes using these hive plots or these circuit diagrams, these graph matrices, or these network visualizations. And so historically, there have been different types of connectomes. We have the anatomical connectome, which is the chemical or electrical connectome. So chemical, again, is synaptic connections. Electrical is gap junctions. And those have been well defined since the late 70s. Then there's the extrasynaptic. So these are things going on outside the synapse or near the synapse. So you have neuropeptidergic connectomes and things like that. Again, these are just things happening in the C. elegans brain that contribute to the connectome, and we can characterize these sorts of things. Then we have gene-protein expressions. That's yet another set of data. This is the gene and protein expression that's affecting the connectome, either as individual cells or the connections between cells. And then the functional connectome, which of course in connectomics is distinct from the structural connectome, which we might classify as anatomical. So we have this list of historical publications. We start with Albertson and Thompson in 1976. We go to White in 1986. We go to Durbin in 1987. Hall and Russell in 1991. Then we go on to Varshni, Cook. Varshni did a gap junction connectome. Cook did male connectome, or actually I think they did both the male and the hermaphrodite connectome to show the differences between the two. So there's Cook 2019, Cook 2020, and then Britton et al. 21, and Whitfleet et al. 21. The Whitfleet reference is interesting because it involves larval development. It involves getting a connectome for the larval worm, a chemical connectome, and looking at how the synaptic connections change across larval stages. So we've used this in a paper already in our group, and I know that Jayadroth is particularly interested in this data set. So that's those are the anatomical connectomes. Those were the first connectomes to be defined. The next set of connectomes, of course, comes from out of White 1986, and it's the functional connectomes. These are kind of recent, where you have these references in the 2020s, and those are interesting, and they have data sets associated with them. Another set of connectomes is the extrasynaptic connectome, which also comes from White's work in 1986, as well as Albertson and Thompson. And these were they started collecting these in 2013, Serrano says. And then, of course, we have Gendriel, Atlas and Hobart, Bentley in 2016, Rapoli Sanchez in 2023. Those are all this extrasynaptic. And again, these are things like neuropeptidergic connectomes. So it's a very different type of connectome, just based on that criteria. And then, of course, we have gene protein expression. Altoon 2009, they proposed sort of this first draft of the connectome, and then you have Taylor, Yemeni, and Eats 2023, and those are all kind of thinking about the gene expression and protein expression complements of each cell in the connectome. And so they've done some interesting things in how to view the data. They have the data sets here, so if you're interested in the data sets, you can get access to them. And so this is all sort of provided by OpenWorm. And the whole point of this is to make this easier for people working on these types of projects to get access to the data. Okay, the next thing I want to talk about is a fun blog post. This is from bluejade.com. This is a Physarum explanation, and this is how, they're thinking about how to do biological modeling, or maybe more to the point, organic simulations. And so they're thinking about, this has a long history in computer science. If you go back to the Boyd's algorithm by Reynolds, or you go back to some of the early computer graphics work, is based kind of on this organic looking thing. And we wanted to kind of move away from these highly pixelated, highly artificial looking graphics, and have things like flows, and have things like Perlin noise, and have things that look like life. So you had the Boyd's algorithm, you had some early A-life algorithms, you had swarming and flocking simulations that built upon Boyd's. You had other kinds of organic simulations of, like, axonal growth and other things. So these are all things we can implement in algorithms. They look like life because they're based on living systems. And so this just kind of talks about how to build algorithms to do these kind of interesting organic simulations. And one of them is modeling the slime mold Physarum. So that's what they're going to talk about in this article. So the purpose of this article is to explain techniques that enabled me, the author of this, to make simulations like the one below. So if you look at the simulation, it looks like it's a flow. It's a Physarum. the slime mold that's propagating or flows in here. And again, we're just, there's this qualitative aspect of it that, does it look like life? Does it look like something that is a living system? And, I guess you could say yes, because it's your intuition. You've seen, living systems, you've seen slime mold, you've seen bacterial cultures, you've seen different biological processes playing out in front of you, so you can tell if something looks organic or lifelike or not. And so this is what they're kind of getting at. So it's very, it's artistic, it's scientific, it's also very intuitive. And so I don't mean to say that this is like, we can use all these techniques for scientific simulation, but they do have some value in scientific simulation. So we will focus on algorithmic techniques for artistic purpose rather than scientific meaning. So this is the first example. This is the Physarum algorithm from Jeff Jones, and this was produced in 2010. Jeff Jones presented a simulation algorithm that reproduces the behavior of organisms such as Physarum, polycephalum. It is explained in this paper, which is this paper, Characteristics of Pattern Formation and Evolution and Approximations of Physarum Transport Networks. And so just as a sort of to get a sense of what the results look like, they look like this. These are Physarum colonies, but they could just as easily be like blood vessels or cells in an array. So, we don't, and we could describe this in a lot of ways. If you just show this and don't put a label on it, people might make different guesses as to what it is. But it's lifelike in the sense that it looks like something that could be produced by a living system. So the general principle behind this kind of simulation is that particles, or what we might call agents, move around, leaving a trail behind them, and then try to follow the trails they detect. So this is like very much like a ant colony optimization algorithm, where you have ants and they're moving around the space, and when they move, they leave a pheromone trail. And if they're successful at, say, finding food, that pheromone trail is followed, and then the successful trails are reinforced by other agents, and then you end up with this network of trails that are reinforced because they lead to something, they do something. And of course, because the, say, maybe the locations of food in the environment are random, there's this stochasticity to them, as well as the behaviors of different agents following either the original trail or blazing their own trail. So there's this stochastic component which makes things look lifelike. So this is just the simple algorithmic principle, though, which is all you need are some agents. You put them on a grid, and then they just move around, and then they reinforce each other's behavior. So they follow simple interaction rules, and then from that you get these different patterns that emerge, that form, and they look lifelike because they follow these kind of principles of maybe living agents doing things and behaving. So this is an interactive explanation of a single agent without representing the trail update. So you can see this. This is the simulation here, where it's kind of like the agent is moving, and it's moving to these different positions, and you can see that this is unfolding on a grid, so you get this pattern that emerges as it moves around the grid. And then you have a lot of agents in parallel doing the same thing. So, detailed description of this algorithm is a large number of agents moving in a two-dimensional space. At each iteration of the algorithm, agents deposit a trail on an image called a trail map, which is this green and light green and white array up here, just by adding a value to the pixel of an image. In the above illustration, each cell represents a pixel on the trail map image. So, of course, the agents have to have some two-dimensional position represented in X and Y, and then an orientation, which is a heading or an angle of movement. So those are the agent attributes. And then there are these different steps for iterating the algorithm. So there's the sensing step where the agent looks ahead at 3 places. The distance is defined by the parameter sensor distance. So each place has a distance that's estimated. The side angle has a parameter called sensor angle. And then the agent detects the trail intensity at these 3 positions or pixels. So there's this prediction that's made. There's this distance that's calculated, this angle, and then there's this sort of estimation of what happens if this action is taken. So that's the sensing step. Then there's the rotation and movement step, which is where there's an evaluation of making a move and then executing it. So this is where you're getting this actual action, and it's not just an estimation, now it's actually doing something. Then you have this deposit step where the agents add a value to the trail map at their new position. So this is like depositing a pheromone in the ant colony optimization algorithm, but instead this is just kind of leaving a memory trace in the environment. They call this stigmergy, or this sort of extra agent memory. And so it's a way not only to deposit information for the agent itself, but other agents can follow that information out. The fourth part of this algorithm is trail diffusion and decay. So this is where the trail after deposit becomes diffused as time goes by, it decays. And so, we don't want to keep this trace around forever. We want to keep it around for a little while because the environment changes. And if you keep every trace, first of all, you can't distinguish between any one valuable trace versus any other trace. And secondly, sometimes those traces become irrelevant over time. So we don't want to have false information in the environment, hence the diffusion and decay. And remember, we're implementing this with a computer, so we're limited by how things actually happen in nature. We have to use common mechanisms that we can represent in a computer. We can represent diffusion rates and decay rates pretty easily in a computer, but implementing something else, I don't know what it would be, that life might do, that's a little bit different story. So this is real interactive implementation with a few agents. This is showing the sensor distance, sensor angle, rotation angle, and move distance. So if we increase move distance, we get this different behavior. We get this faster behavior. It's more frenetic looking. Sensor distance as it increases. They all segregate out on one part of the grid. Sensor angle, if we decrease that, we get this, if we increase it, we don't appreciatively change the behavior. And then rotation angle, we change the behavior a little bit, but really sensor distance is the thing that's really affecting this. And you can see that now I've formed, with these parameter values, I've formed the single lane of the agent moving and this sharp gradient of a signal. And so this is really interesting how you can change these macro parameters by changing the parameters for each agent in parallel. And so that's okay. So then there's the second one is 36 points by Sage Jensen. This was from 2019 to 2022. This is a more complex version of this first algorithm where it shows these different varied behaviors. And so it uses a single algorithm, and each point represents different parameters, which result in different behaviors. A point corresponds to a behavior obtained with 20 parameters. So mathematically, the project shows 36 points of R space 20. So that's what this is. And then, again, we have this simulation, same thing. Then the third one is the BlueGA implementation, or the author of this post. And so now they've released this implementation of the previous algorithm. This is on GitHub. under the account Physarum, or under the BlueG account, but the repository Physarum 36p. So the code is available for this. Basically, you have some make files and then this README here, which tells you how to run this program. And yeah, so that's, and then this is their implementation. And this implementation produces a result for 22 different points, each having 15 parameters, and this is the simulation. So you see these sort of, it almost looks like, so it's like these stochastic events are forming these trails, and may look like blood vessel formation, may look like bacterial colonies growing and merging into one another. It might look like some other diffusion limited aggregation process. Now it kind of looks like the flow of liquid or some sort of thicker viscous fluid. These are all really interesting things, and again, it's very qualitative because we're not measuring the extent of these different structures or characterizing them with any sort of sophisticated mathematical model. We're just simulating with these parameters and seeing what the result is. So it gives them information. They give some information about real-time setup, diffusion and decay algorithm, depositing things on a trail map, and then some reflections on the differences between the different variations on the model. You have these color experiments, velocity effects, playful interaction ideas, and points mixing. So these are different versions of the algorithm. You can play around with them to see if they do interesting things. And so that's this is the interactive Physarum repository, which kind of shows some of the results, and then some other experiments where, as this algorithm stands, it's workable. You can get it to work, and it can get it to do a lot of interesting things, but there's also a lot of room for experimentation and sort of teasing out the parameters and figuring out what works even better or what produces new and novel results. So this is all very interesting. And again, it's this sort of qualitative aspect of simulating life, but I think it has a place in informing what life is or how life might do things in lifelike systems. And it isn't a reductionist sort of approach. It has it has all these different things. Of course, we talk about mechanics, we talk about fluid dynamics as part and parcel of, like, say, cell biology or morphogenesis. And I think this underscores the point of that, because to get things to look lifelike, you need to have fluid dynamics, you need to have different types of mechanical effects, you need to have interactions, you need to have then, of course, the cells themselves and their own properties. This kind of collapses all that down to a simple interactive simulation, but there are all these different, we have, say, like we consider the equations of fluid dynamics, the equations of mechanics, things like that, for a reason, because we need to be more precise in when we backwards engineer some of this stuff and try to figure out the mechanism. Although this is not, I would say that there is a limit to how much how we take the reductionist view of this. So here's some additional resources, and then hopefully I think this has been very inspiring for our group. So if you have any questions, this is by Etienne Jacob, and you can find this at bluej.com, Physarum explanation.